The overall goal of the following experiment is to culture non-enzymatically disrupted sterile breast tissue containing areas of in situ ductal carcinoma or DCIS lesions to engender the spontaneous formation of mammospheres in vitro. This is achieved by partially submerging small pieces of breast tissue containing ductal segments in a minimal volume of serum-free medium to provide an oxygen and nutrient gradient within the organoid. Next, the organoids are cultured without disturbance until the epithelial cells and fibroblasts emerge from the open ducts and attach to the tissue culture flask. Ultimately, the epithelial origin of the resulting spontaneous mammosphere formation can be confirmed by both direct microscopic examination and immunofluorescence staining.
The main advantage of this technique over existing methods which use enzymatic dissociation of the breast tissue is that our breast DCIS organoid culture maintains the biological context of the ductal microenvironment without disrupting the in vivo cellular interactions.
Although this method can provide unique insights into the biology of breast ductal carcinoma in situ, it can also be applied to other types of pre-invasive neoplastic lesions such as pancreatic intraepithelial or prostatic intraepithelial neoplasia.
When performing this procedure, it is important to take care that the tissue remains sterile during the transport between the surgical suite and the tissue processing area. Furthermore, the instruments and reagents used for the tissue processing should be sterile to prevent contamination when grossing the tissue.